So our first presenter today of our Gateway Showcase is Chengxin Zhang, who is from the University of Michigan, and he's going to be talking about I-TASSER, and then he'll be talking for the next half an hour or so, and then we'll be hearing from Perri of Chem Compute. So, Chengxin, go ahead.
Okay. Hello, everyone. I'm Chengxin from Zhang Lab. I'm the current webmaster of I-TASSER. Today, my talk will be divided into 3 sections. I will talk about how I-TASSER can predict protein structure and function, and how we use the XSEDE resources to accelerate this process. In the second section, we will introduce how we can submit jobs to the gateway, and when the result comes back, how to interpret it. And finally, we will introduce in the final section a small trick to improve the quality of the I-TASSER models. But before we jump in the principle of I-TASSER, first let's have a very small review on what is protein structure and function prediction problem. In this figure, on the left-hand side, we are showing the protein sequence, the amino acid sequence of the protein. Unfortunately, only knowing the amino acid sequence of the protein cannot tell us what is the biological role or what is the specific task this protein can perform in a living organism. That's because usually for a protein to carry out biological tasks, they need to fold into specific 3-dimensional structures as shown in the middle. The process of predicting the protein structure from amino acid sequence is called structure prediction. In our example, you can see that in the middle of this structure, there's a pocket. And this pocket is responsible for binding small molecules and other proteins. As illustrated on the right panel, our this protein can bind the ATP small molecules and transfer the phosphate group from the ATP to the tyrosine amino acids on another protein. This will convert the ATP into ADP, and the tyrosine amino acid will become phosphotyrosines. The process of predicting the protein function is the function prediction problems. So even though protein structure and function is important, to experimentally determine the structure or the function is by wet lab biologists is very time consuming and also expensive. That's why we developed the I-TASSER gateway to automatically predict protein structure and function. We will show their working principle in next slide. In this slide, on the left dash box, we are showing the protein structure modeling process of I-TASSER. And on the right panel, we are showing the function modeling process of I-TASSER. Let's first talk about the structure prediction by I-TASSER. Starting from an amino acid sequence provided by a user, we use threading to search this sequence against the PDB protein structure database in order to identify template structures that are structurally similar to our query sequence. From this template structure, we take the structure fragments and assemble them in a structure assembly simulation using Monte Carlo simulation. This structure simulation process will generate thousands of different protein structures for the same sequence. In the next process, in clustering, we grouped the different structures from the structure assembly simulation into groups into clusters and by the structural similarity between the different structures. For one big cluster, we take the centroid of the cluster. Since this whole structure simulation process is very time consuming, we only take for each amino acid 2 atoms to represent it instead of the full atoms. Since we need to get the full atom model in the end, in order to refine local geometries and to reconstruct the full atom models, this cluster centroid undergoes structural reassembly and full atomic refinement in order to get the final model. So this is the structural prediction process of I-TASSER. And using this final model, our in-house program, Coach and Cofactor, will search this final model against Bioinformatics database, which is a database of protein structure with function annotation. Coach and Cofactor will, based on the similarity between the database entry and the final model, transferred the function annotation from the identified structure to our query sequence. And this is the function prediction process of I-TASSER. So the most important and also the most time-consuming process of I-TASSER pipeline is this part, structure assembly simulation by Monte Carlo. Therefore, in the next slide, I'm going to go a little bit more detailed on this process. The structure assembly simulation is guided by a composite energy function, which contains 3 different kinds of energy terms. The statistical energy terms are derived from experimentally solved protein structures deposited in PDB database. The template-based restraints are extracted from the template structures obtained by threading the sequence against the PDB database. And we also have user-specified restraints. To calculate the total energy for one structure and to move one structure to another structure is very time-consuming process. That's why we need that's why we use XSEDE resources to accelerate this computation. Here I'm showing the architecture of the I-TASSER gateway. A user of the I-TASSER gateway can submit a protein sequence to our web server. We copy this web server to our own cluster in our lab, which is labeled as Zhang cluster layer in this image. In our own cluster, we will pre-process the input file provided by the user and then use rsync to copy the input file to the commit cluster provided by XSEDE. At commit, we have a cron tab program which periodically track the input files and submit the jobs to the computing nodes of commit. After the structure modeling is finished at commit computing node, we use rsync again to copy the output files to our Zhang cluster, where we prepare the final web page output. This output data is deposited in our central database, and we showed the final output results in our web server. Finally, we will send an email to the user to notify them that their job is finished. Thanks to the commit cluster provided by XSEDE, we can now handle a large amount of protein sequences provided by the user. To put this in perspective, I am showing a plot of I-TASSER usage since the establishment of I-TASSER Gateway in October last year. So every year on average, we have around 5,000 to 6,000 protein sequences provided by user every month, which which means on every workday, we have close to 200 protein sequences provided by the user. And due to the commit cluster, we can relatively easily handle the provided protein sequences. So I-TASSER is a popular web server for structure and function modeling. So in the next section, I'm going to introduce how you can submit your jobs and how to interpret the output results. Here is the input web page for I-TASSER Gateway. Since it may be a little bit difficult for you to see in the slides, let's go to the web server input page. So here is the I-TASSER input page. In this text box, you could provide your protein sequence and also your email. Since I-TASSER, even with the commit cluster, is still very time-consuming to perform. That's why we currently only mainly serve the academic users. So if you have academic email, you need to register here, where you provide your name and your email, and we will send a and we will provide a password for you to submit your jobs. After you provide this, you just click Run I-TASSER, and your job will be submitted. So usually, it will take a few hours or even a few days for your job to finish, depending on the length of your proteins. After your job is finished, you will get a web page output where we provide the sequence, the template, and also the final model and function prediction. Since the font is getting a bit smaller in the web page, let's switch to the slides where we will introduce the output. This table shows the top 10 templates used by the I-TASSER simulation. This row is the secondary structure predicted for our query sequence from position 1 to position 120. The second row shows the amino acid sequence for our query. Each row underneath is one template identified by threading my query sequence against the PDB database. Here, for the template protein sequence, if there's an identical amino acids to our query, we use color code to indicate this to indicate the identical amino acids. So in the first column, we have the PDB ID for the template, and for the second and third column is the sequence identity between the template and the query. We can see that for most templates, there will be gaps. In other words, one template does not necessarily cover the full length of the sequence. That's why we have a column for the coverage of the template. And we also provide the alignment score or the Z-score between the template and the query sequence. So the next and also the most important panel is the structure model, the final structure model predicted by I-TASSER. Usually, we will predict up to 5 models for each query protein. I want to draw you to attention to the estimated TM score. So, we provide estimated TM score to quantify what is the estimated structure quality of the predicted structure model. TM score range from 0 to 1, and with higher TM score indicates better structure quality. And usually, with TM score greater than 0.5, the structure modeling is confident and trustable. Also, the structure models are already ranked by their quality, so we usually recommend that you use Model 1 for your subsequent biological study. Since we already mentioned before that since I-TASSER use structure to predict the function, we show in the next panel the function predictions for our query structure. Here we are showing the ligand binding sites, or the amino acids on our proteins that could interact with small molecules. In each row, we showed 1 templates that are identified by code or cofactor that is used for ligand binding amino acid identification. So this column shows the ligand binding, shows the index of amino acids that can interact with small molecules. Such amino acid is colored in blue on the left panel, and their index is in magenta font. We also provide the name of the small molecules that might interact with the target proteins, and this small molecule is shown in green in the left panel. You can also download the structure of the ligand of the small molecule superimposed into the target protein structure. So to show the functionality of this panel, let's go back to the website output. If you scroll down, so in this panel, on the left, you can rotate and also to zoom in or zoom out your protein structure. When you click different options here, it will show the prediction result using different template structure. And in the next section, I will introduce how to improve the quality of I-TASSER models if the protein is very long. So for such long proteins that has like hundreds of amino acids, they are usually consist of multiple domains as illustrated on the upper left panel. So for this specific example, where we are showing the sequence with 480 amino acids, it has 2 parts. It starts with EGF-like repeats, color in blue, and it is followed by 2 discordant-like domains. If you submit the full sequence to an I-TASSER web page, the top 10 threading template alignments returned by the I-TASSER will look like this. On the so each row is 1 template, and in the green box, all the templates covers the query sequence pretty well. But on the left blue box, more than half of the templates, they don't cover the query sequence. In this case, we can we can estimate that there's a domain boundary between the blue box and the red box and the green box here. So if this is the case, you should split the full sequence and submit the sequence for the inside the blue box and in the green box separately to model the structure, individual domain structure using I-TASSER, and then reassemble it after you get the individual domains. So if you are not an experienced user and you have difficulty interpreting this alignment, we have automated this domain boundary prediction process by implementing two complementary web servers, ThreadDOM and ThreadDOM EX, which automatically use threading alignments for domain partition. With that, I will finish my talk, and I will thank XSEDE for organizing this talk and also providing the essential computational resource for the gateway. And I will want to, so Zhang is our PI and developed the first version of the I-TASSER, and Ambridge and Jianyi was the previous webmaster of this gateway, well, of this web server. Mortuza is currently developing the next generation of I-TASSER gateway, which will have better structure modeling result. Thank you, everyone.
And thank you, Chengxin. We have had one question come up while you were talking. Mona asked, how big are the data files for the user input and output?
So usually the input is just a very small text file, less than one kilobyte. The output is much larger. So if we don't count intermediate intermediate results, just the final output is at least 20 megabyte. So
okay. So it's not huge, like as much as it looks like it would be with all that information there. Then.
Well, yes and no. So the most data are generated during the simulation process, where we have thousands of protein structures from the simulation, but we don't save it for the final output because the disk space is limited. Otherwise, there will be like hundreds of megabytes for each protein.
Okay. So Moni can let us know if you have further questions to that. Another, Joe Allen asked a question. He says, you mentioned that Comet periodically checks for new data and submits jobs when found. How is this done? Is it something like a cron job and template batch script? I don't know the lingo.
So we are using crontab. So we have a crontab that's set up in the panel of Comet that runs every 15 minutes and see if there's some job that needs to be run.
Okay, I'm curious. Another question. So we got a thank you from Joe that answered his question. I know that one of the things that was particularly remarkable about I-TASSER when it first joined XSEDE and started using those resources was that it dramatically increased the use of XSEDE resources for science gateways, which is of course interesting to us, being all gateway, supporting gateways. And I was curious to know if you were anticipating that, because I understand you moved to Comet because you couldn't handle the capacity on your own servers. So I'm just curious if you were anticipating that kind of volume once you expanded.
Yeah. So actually, we do anticipate there are that many users. So as we mentioned before, there are close to 200 proteins submitted by user daily. So before we use Comet, can't finish it. Uh, we cannot finish the protein within a week very frequently.
Mm, yeah. So you realized that was a limitation in terms of
Yeah,
capacity. Yeah. Yeah.
yeah.
Another question came in from Randy Highland. He asks, do you know if I-TASSER has ever participated in the CASP contest, CASP? I don't know what that is, but
Yeah. So I participate in CAS 8 to CAS 12 under the name Zhang server. So since CAS 8, it is the so for the last 10 years there was 5 CAS sections, and in 4 of it, I-TASSER is the ranks as the best structural modeling server. There was once where Quark server, which is another server developed in our lab, was the first.
Randy says, thanks. So with that, I think we don't have any other questions coming in unless someone wants to jump in. Otherwise, thank you very much. And Mona, also thank you for your presentation. And thank you for me. So with that, we'll switch to the other gateway that we're going to showcase today. And I suppose if other questions come in, we can always pass them along to Chengxin as they, because we know where to find him. So now I'd like to introduce now Mark Perri, who is from Sonoma State, and he has been developing the Chem Compute Science Gateway with some help from our own developers here at the Science Gateways Community Institute. So take it away, Mark.
Thank you. Yeah, I'm Mark. I'm an associate professor here at Sonoma State in the chemistry department. And so today I'm talking to you about our gateway, and our gateway is maybe a little different because it's really geared towards undergraduate students in chemistry. So Sonoma State is one of 23 Cal State universities. So we're a public university. We are a PUI, which is a primarily undergraduate university. So that's why I geared this towards undergraduates. A medium-sized school, about 9,000 undergraduates and 800 grad students, but our department has no grad students. We're only a bachelor granting institution. And as a small or medium-sized undergraduate institution, we really have almost no resources for computational chemistry to buy almost anything, and so that motivated me to make this gateway. So our gateway is chemcompute.org, and you can run 2 packages on it. So Games is a quantum chemistry package. It does quantum calculations, which basically looks at where the electrons are in molecules. And then Tinker is molecular dynamics. Molecular dynamics is for the motion of molecules. So you can see molecules moving around inside of a box. So why would I make a gateway to educate our students? The main reason is that trying to implement computational chemistry, I found a lot of barriers here. And having a gateway makes it just a lot more simple and solve these barriers, as I'll talk about. Another reason is I have this dream that everyone's in lecture, we're talking to our students and teaching them something, and then you could just bring out your tablet or your phone or your laptop, and everyone in the class could be running some sort of job. It's nice to have it on a website because students can access it from home or from their dorm room, and they could run a job that might take a few hours, and then it's ready when they come to lab, and you can spend time visualizing the data instead of just waiting for the job to run. It's nice that we can be accessed from other countries. I like that aspect of it, helping out people who don't have resources themselves. And a website really is familiar to students. They love to do Facebook or whatever they're doing. And so something about a website puts them at ease rather than running an actual package that's installed on a computer. They have a lot of nervousness around physical chemistry and math and computers, and so this helps maybe a little bit with that. And then Gateway makes it easier for faculty to implement these labs. I'll talk about that in a little bit. So one of the reasons I want to use computational chemistry in my teaching of undergraduates is that a lot of these chemical concepts, a lot of times they're explained with equations. And so here's an equation for a 2p orbital on an atom, just basically tells you where the electron is in space. And so we show this a lot, and we say, here's, there's an angular part to this equation, and there's a radial part to this equation, and can't you see what the orbital is going to look like in your mind? And the students don't respond to that well. So if I can have them pull up the Gateway and look at the p orbitals on an atom, something very simplistic, it actually helps them a lot in their understanding. And this is really one of the fundamental building blocks for chemistry. So our usage, because we are an undergraduate website, we're used by classes. And so our usage is really seasonal. It peaks, in the late fall or late spring, when these classes, the semester is going and they get to this sort of place in their classes. And so we don't have really many users during the summer session or winter session. And so it's kind of this odd thing where there's nothing going on, and then all of a sudden everyone logs on and does a job, and then we don't see them again for a couple months. Because we're in classes, we tend to get hit with mostly just really small jobs. We don't have jobs that run for hours or days or take up, 18 nodes or anything. What people want is for their students to run a job and then look at the output. And so you can only run jobs that take just a few minutes. And so we can do like single core jobs, just a few minutes, just really simple things. It don't require a lot of computer power per se, but because there's a lot of users, maybe simultaneously, we need resources for that. And then some students do use it for research, which is I'm trying to implement more, trying to promote it more. We had a student last year use it as their honors thesis. They didn't have any resources, and so they did their whole honors thesis on the website. Some students log in and do some jobs for the research. And I really like that. The research might be a minority of users, but it gets to be an equivalent amount of SU units because their jobs tend to be longer and more complicated. So we're a little small. We're just starting out. Since fall of 2016, we've had about 25,000 jobs run, and I try and track it from around the world. The US is our main user of jobs, but also Germany and Australia, I see them run jobs in their classes, and then some other countries use it for some research. So we're trying to get global, and we're really trying to increase the number of users and the number of jobs. So to make the website, I programmed it. This version of the website is in Django, and so everything runs using Python. Django runs Celery, which is its task queue. And so whenever you run a job or you want to get some data, it'll run Celery to process the data or to submit the job. And then we use a message queue, RabbitMQ, that shuttles messages back and forth that this job is done or this job is queued or whatever is going on. And then everything has a database in the back end. All right, so that's the website. Let me tell you about the barriers we faced at a primarily undergraduate university. One of the barriers is just hardware. And so it's actually difficult to get computers to run anything on, even small jobs. We don't have like a university cluster like a larger university does. You can go in the computer lab, but that's really inconvenient because having IT install software just becomes a nightmare. So it's nice that we can use the XSEDE network. We can use Comet and Bridges to run longer jobs, more serious computations. And then we have an allocation on Jetstream, which is nice. We get a lot of these really short jobs, one core, just a few minutes, maybe a couple hours. And Jetstream is nice to do those. And we also use Jetstream for guest accounts because it's just a virtual machine and there's not much for them to ruin. Jetstream, we run through OpenStack. And so, I just run sort of a small node all the time. And then if I get some jobs coming in, there's a script that runs. And if it sees that there's a lot of jobs queued, it'll launch a couple other extra nodes. And so we can scale up or scale down based on demand, which is important because our demand is so seasonal and it varies throughout the day. And then it's nice to have a few XSEDE resources because, our jobs are actually kind of time critical, because if you have a lab at 1 p.m. on Wednesday and you get in and you find out that Comet, is under maintenance, that just ruins it for you. If you are doing something for research and there's maintenance going on, you can queue your job and it'll run when the maintenance is done, come back in a couple of days and that's fine. But the students are in lab, they're there, and they need to be able to run their jobs right at that second. So the other barrier I face is the access to software. You can buy really nice software packages for this, for computational chemistry. It's really easy to use, but it's expensive. And we just don't have the money to do that. And I think there's a lot of institutions like us that don't have the money, but have the willingness to do it. So there are free software like games that we run, but free software, is not easy to use. You have to make an input file that's a text file, and students really don't like that. And so what I've tried to do is bring in some tools to let the students set up their job pretty easily for them. And so I'll take you through a typical run that we do. We look at benzene, a molecule, very famous molecule for chemistry. And we're going to calculate basically where the electrons are in this molecule, which tells you how the molecule is going to react. And so first thing you need to do is make your input file. And so to do this, all you need to do as the student is to draw a benzene molecule, which hopefully any chemistry student can do. And there's even at the top of the screen, there's a little benzene button. So you can just click it and drop down your benzene molecule. You can also search for a molecule. We have it hooked into a couple databases. If you type in benzene, it'll go to this database and find it and bring in the structure for it. The input file is in 3D, so you need to have XYZ coordinates for your atoms for the molecule you want to calculate. And so you can transfer from the drawer, which is called the JavaScript molecule editor on the left, in 2D into JSMol, which is a 3D molecule viewer. And these are all open source that I've incorporated into the website. So once you have it in 3D, you're halfway there because the input file just has your XYZ coordinates. And then the next thing you need to do is all your settings. And so this is part of a work in progress. I'm trying to make this a little better for the students. Basically, all you do now is click all your settings. A lot of settings of what you want to calculate, what the method of calculation you want is, if you want to do it in some sort of solvent, what the charge of your molecule is. And I had a dream of what this should look like, and I just need the time to program it in, or maybe the skills to make it happen. But once you click all these and you click Create Input File, the computer, your client through JavaScript will translate all of these settings into all the keywords that are needed in the input file. Then you have to submit your job to the server, and there's two ways that we've done that. We've used Arvata through Python. There's a Thrift translator that'll translate everything into the Arvata API. And Arvata is nice because there's lots of support from the team, and it's easier to use because whether you're using Torque or SGE or Slurm, it's got stuff built in there for you already. And so all you need to do is make your API calls with the Python. But the difficulties I've had with it is there's no allocation model. You can't say this user has this much allocation. And so the problem with that is it's easy for undergrads to just submit a huge molecule that would take days and days to run, and they could run it on a full node or whatever, and all of a sudden they burn through your allocation. And so I limit the classes to how many SUs they can use and how many jobs they can run. And then there's no intermediate output. It's nice, it'll submit the job and get it back for you when it's done, but there's nothing in the meantime to show the student. So I've also used SSH, which is nice because it's fast and simple and straightforward. You just send a script to the computer and tell it to queue it up. With SSH, we can have it get the intermediate output back in time using rsync, and so the students can view it as the molecule is being calculated. But of course, the drawbacks, you have to write everything yourself. And if you have too many SSH connections, that's always bad. And so we use a lot of multiplexing to solve that. And then to keep track of the jobs, there's a database, MariaDB, which I think is just the same as MySQL that we use. And then periodically, when the student is viewing their output, Celery will run in the background, and Celery will ask using SQ or whatever commands to see the status of the job. When the job's done, it sends a message through RabbitMQ, the message passing system, that will tell the gateway that the job is done and how long it ran for and all the stuff for it. It's easier to use RabbitMQ than to shuttle messages through SSH because it doesn't have a lot of the overhead that SSH does, and you can have a lot more connections simultaneously with Rabbit. And then in our internal database, we have a user database, and the profiles, we have how many allocations they have, and I can change it per class based on their use. All right, so once you get your job submitted and it's done, you get output. But what I've found is that the students don't like to wait for the output. And if your output even takes 5 minutes to get back, the student will resubmit the job because they're just like, oh, nothing's happened. So, and all of a sudden they've submitted 10 of the same job. And so I'm trying to give them something back immediately. And so it'll start sending back the output file. And so you'll see at the top of the screen, this job has been submitted to this node at this time, and it starts scrolling through the output file at the top of the screen. So they can see that something is happening. And that's important for them. And then they don't like to look at the text, so they like to see the text that it's going, but they don't like to look through the output file to see any information. And so I've tried to make all the information parsed and available on the screen for them. So to get intermediate output, the easiest solution that I've found is to launch sort of a helper script in the background on the compute node. And all it does is it looks at the output file and sees if it's been modified. And if it has, it parses the output file and it sees what the newest prediction of the atomic coordinates are. And so it can send those coordinates back to the computer. And it's better to send just a part of the output file, because if you send the whole thing, all of a sudden you're sending 1 megabyte, 2 megabyte, 3 megabytes, and it just bogs everything down. And then when the job is done, it sends a signal and it'll download the full job, full output. So to visualize it, we use JSMol. This is an open source molecule viewer. It's really nice. It'll read the whole output file and do visualization. So the molecule here is benzene. This is the output. And you can see the red and the blue. That's telling you where the electrons are in the molecule. And where the electrons are in a molecule is basically all chemists care about. It tells you everything about how it's going to react. And so it's very important for the students to visualize this to help them in their understanding of chemistry. And they can compute other things, like this is an infrared spectra on the right. This is where it absorbs infrared radiation and where it doesn't. And so there's a variety of packages we use to parse the input file or the output file and then turn it into a graph. And so we use matplotlib on the Python side, and that'll turn it into a graph that it can download in JSON format to your client. And then the JavaScript, the D3 viewer, will view that and it can show it as a graph. And then a lot of times the students' runs don't work for a lot of reasons. The most common reason is that they get the multiplicity wrong. So you have to tell the computer what the spin of your molecule is, and this is very important to the calculation. And so I've seen that there's maybe like 10 common errors that the students always get. And the students never want to actually read the real error or look at the output file to see the error. So the Python client, the Django, it'll read the output file, and if it sees these particular errors, it'll translate them and it'll say, hey, make sure you check your spin or multiplicity because one of those is not right. The charge is not right on the molecule. You need to resubmit. So for the molecular dynamics, it's really just showing where and how the molecules move in a box. And so this is nice for the students to see. They love looking at the picture or the movies of it. And so it downloads it sort of frame by frame, and the JavaScript client will put the frames together and display it as a movie. And it also displays some information about energy versus time. So the third barrier that I had, so we had hardware barriers, access to software. The third barrier is just access to what to actually do. It's hard to find lab exercises for computational chemistry. And when you do, they're usually for a specific package, and you might have a different package. And so on the website, I try and incorporate some exercises that are built in, and they work with the website. And so there's instructions on the screen. So because it's a website, I have control over where everything goes. And so just on the right-hand side of the screen, it says, draw this, click this, and it's got questions. What do you see? What's the symmetry? What does this remind you of? And so the whole lab is basically built into the website. So the faculty doesn't need to go and find something, write instructions or translate instructions to what they have, and then write questions and have instructions. It's all right there. You can have there's pre-lab questions, there's a link to background theory that the students need to read, and so it's really geared towards just a student coming in, going to the link, and starting the experiment. So I've used this in class a number of times, and I have some personal observations that I've seen. It actually works way better to have them use the gateway than anything else I've ever used. We had one license of this computational package, and so everyone sort of stood around one student using it, and that was terrible. And then I tried to do games by itself on the terminal, and that was terrible. And then now using the gateway, I see that all the students have completed the experiment, which is a big plus. I used to have to let them download the output files when they couldn't do it, and then they would just look at it so they could get data. But all students completing experiments actually is a big thing. And the website, they've told me that it makes them feel a little more comfortable. I get less complaints about, I'm not a computer person, I shouldn't have to do this on a computer, I don't like math. Having it on a website does seem to put them at ease. At the end of the lab that I do, I have them do their own molecule. I say, you pick a molecule and pick your calculation and do whatever you want. And a lot of the students do a molecule that they have been synthesizing or analyzing in research. And that's nice. I like to bring that into the class. But some of these calculations require several hours when they get to pick the molecule. It's all of a sudden a long calculation. And so the fact that it's on a website is nice because they're not limited to the time in the computer lab. They can go home and wait for it to finish and then pull up the job output and then write that up in their lab write-up. And I've seen a lot of misconceptions. The nice thing about the gateway is that every student gets to do it. You're not limited to a certain number of licenses, and you got to have 2 or 3 people using it or 12 people using it at once. And so I can talk to them individually, and visualizing molecules, I've exposed a lot of misconceptions that they have. And this has helped me in my teaching. So I've asked them some survey questions before using the website and after using the website. And so it does seem to be better for them. They have said that they feel more confident using computers to solve chemical problems after using the website. And my survey scores, where they evaluate us at the end of the semester, increased in 2015 after incorporating this into the class. They said they participated more actively learning, that I stimulate more interest in the course. So I've enjoyed that part to helping them learn chemistry through this computations. And for funding, I've tried to get funding. It's hard for me to find the appropriate agency to fund this because it doesn't seem to fit in a lot of cyber infrastructure type grants because it's not, like cutting edge research. We're just trying to provide a lot of access to students who don't have access to these programs. But the XSEDE has been great. Access to their systems has been great because I don't have anything for them to run anything on. So using Comet and Bridges and Jetstream has made all this possible. And the Science Gateways Community Institute has given a lot of support and funding for travel and through the extended developer support program, which has been great. So I'd just like to thank my coworkers, Melissa, Rob, and Lane, are working on developing some experiments. So those will be up there soon. And then the whole team, Sudhakar, Roma, Marcus, Suresh, and Marlon, who have helped me immensely with advice and mentoring and help with Arvata, and some funding from my university for this, and then the gateway allocation. And then they made me put the slide up at the end.
Thank you very much. Yes, I appreciate you putting up that slide at the end. So I put a link in chat for those who have to sign off before we're done, or even after. You can access that for your evaluation. But I first want to thank Mark for that. Was really interesting and easy for me to follow, even though I don't know that much about chemistry anymore. It's been many years since I've learned about chemistry. So I welcome questions from anyone. I had a question which I will ask you while I am waiting to see if anyone else has questions, which was that earlier on you mentioned that students will submit a job in class and it'd be like one node, and they know that it's a small job and it won't take much time. And I was wondering, how do they know how long the job will take so that it'll complete quickly?
Oh, they don't. So the instructions tell them to do certain molecules, and I know those certain molecules will complete quickly the way I've structured them. When they start picking their own molecules for the individual project at the end, their jobs sometimes take forever, because in the experiment we'll do hydrogen, benzene, water, molecules that have 2, 3, 5, 6 atoms, like not a lot of atoms, and then you think that they'll follow that trend, but all of a sudden they'll do a molecule with 500 atoms, and so all of a sudden the scaling is ridiculous, and their molecules take forever. So it is nice that they can let it run overnight. What I've done in the past, if we do it in the computer lab, I say, do something you want, and it won't finish in time. And then the next class comes in, and then they have to kill all their jobs because they can't have the computer being super slow for the business class. So it's nice to have the XSEDE resources that they can run their jobs overnight, because they really have no idea how long these things take until they try them themselves.
Great, thanks. Yeah, that answers that. I was wondering, it was sort of mysterious. I was wondering, any other questions from our crew? Okay, Randy Highland says, regarding backend software, have you looked at using NWChem?
Yeah, there's a couple of free open source software. Why did I use games the first time? I don't remember why I picked it, but I picked games for whatever reason. And games has a certain input file structure, and so I've tailored it for that. And the error messages are specific to games, and everything's specific to games. And so I could use a different package, but I have to translate everything. And so I've just sort of stuck with games, because once I picked it, it's like we're all in with games. It's just everything's already made for games. But eventually I'll look for other programs. People asked me actually for other molecular dynamics packages than Tinker, and so I'm going to look into using NAMD first as an alternative.
Thanks. And then also our other presenter, Chengxin, asked, can you run a system with multiple molecules?
Oh yeah, you can. The games is capable of that for sure. A lot of times people will do a molecule and then surround it by some number of water molecules to simulate it being dissolved in water. And so that's definitely possible with games. But the problem is the way that they build the molecule. It's not easy to manipulate multiple molecules at once. So if you could build a molecule, maybe 4 molecules around each other, but you can't like lasso one molecule and drag it to where you want it and position it. So the positioning and the movement of the molecules is limited, just because of the way I have them build the molecule. But there's an alternative version called like expert version or unguided version, where if you have the skills to create your own input file with multiple molecules in it, you can just upload the input file and it'll run it for you. So you can use it sort of in expert mode for that.
That's neat. Thank you. I'm curious, so based on your teaching evaluations, it suggested that you've been using this actually for, 5 years at least.
The 2012 and 2014 was when I was not using it. So I think I started using it in class like 2015. So there was a little jump in my scores from 2012, 2014 to 2015.
Okay, so then you engaged us about a year ago. So I'm curious if you could speak briefly to the ways that how far you were able to go on your own, and then how it has changed since you've been working with us with extended developer support.
So on my own, so what I used to do is I had a cluster, which is I had these old desktop computers that I just networked together, and so it was technically a cluster. It was like four computers, regular computers, and so I could have our students submit to those using this really complicated way of doing it that I shouldn't have never done. But then now with XSEDE, I've got more power because I can't serve other people with just 4 computers. So XSEDE has been great to expand the usage of it. The CGI hosts the website. I had a problem with hosting the website here where they gave us a very tiny VM, and if too many people logged in, it would run out of memory and crash. And so hosting of the gateway has been great. And then just all the support with looking at Aravada, just looking at how the jobs are submitted and how it's structured. It actually helped me rewrite a lot of things that I do in the website to just structure things better. It's hard sort of, I'm here, when I first started, I was here sort of all alone, just doing things. And when you're all alone doing things in a vacuum, you do things just seem good at the time, but it's kind of not the right way to do it. So to see, the PGA website and see how it's structured helped me rethink some things that I had done and do it a little better.
That's awesome. That's helpful to know. I know that problem working in a vacuum. It's really hard. One last question for you before we sign off. Randy asked, are there any thoughts, have you had any thoughts on using Jupyter notebooks?
Yeah, I have. I've thought about that a lot. So it would be cool if you could have some sort of Jupyter notebook to you could do some sort of processing on the output file. That'd be cool. But I've looked a lot at how to do that and how to run Jupyter notebooks through XSEDE. And I found a couple things, and some things seem to have been started but not finished, and some things I couldn't get to work. And I'm not exactly sure how to do that, how to do that. So I've thought about it. I think it would be cool, but maybe I don't have the expertise right now, and I haven't had time to look into it. And then a lot of times, I'm trying to make. I'm always conflicted. Trying to make it easier to use means sort of having less features, and then having it have a lot of features, but then just sort of be overwhelming for students. And so I worry that having students just typing Python to visualize their data or work up the data might be a little too much for some students. But there's definitely a population out there who could get something out of it. So it's on my list of things to figure out and look at.
Neat. Okay, thanks. Yeah, that's a really good point, not putting so many features just because you can. It's a real usability. It can be a real usability problem.
Yeah.
Yeah. So with that, we're at the top of the hour, and I'm so glad that that all of you were able to join us and hear these 2 terrific presentations.